The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
Hello, and welcome to 507 Biochemistry Online. I'm Dr. Bogdan Fedeles. This video is about pyridoxal 5-phosphate, or PLP, an essential metabolism cofactor derived from vitamin B6. All animals are auxotrophic for PLP, meaning they need to supplement their diet with vitamin B6 in order to survive. PLP is one of the most ancient cofactors, and surprisingly, it can catalyze chemical transformation, such as a transamination, even without an enzyme. PLP is actually involved in a staggering number of biochemical transformations. This video summarizes the most important reactions involving PLP that you will see in 507, and will also show you how to write the complete curved arrow mechanisms for these transformations. Let's talk about PLP catalyzed reactions. As we just mentioned, PLP is the cofactor derived from vitamin B6. This cofactor is very important for a number of reactions. In this course, we're going to look particularly at the transamination reaction. This is a crucial reaction for the metabolism of all amino acids. And we're also going to encounter this reaction when we replenish the intermediates in the TCA cycle, what we call anaplerotic reactions. And we're also going to see PLP involved in reactions in the malate aspartate shuttle that transfers redox equivalents, reducing equivalents between mitochondria and cytosol. Let's take a look at the structure of vitamin B6, or also known as pyridoxine. This is the molecule that we ingest when we get our daily vitamin supplement. Now, in the body, this gets oxidized to form the intermediate called pyridoxal. Notice the aldehyde group here, which is going to be the business end of the molecule. Now, the active cofactor, PLP, it's actually the phosphorylate version of pyridoxal. This requires one molecule of ATP and the enzyme pyridoxal kinase. And we get PLP, or pyridoxal 5-phosphate. The name PLP comes from the initials as outlined here. Now, this nitrogen on the pyridine ring tends to be protonated because its pKa is close to physiological pH, between 6 and 7. Now, for the rest of this presentation, we're going to be abbreviating this phosphate group as such and throughout the course. Now, a related molecule is pyridoxamine 5-phosphate, which we'll see it's an intermediate in the mechanism of PLP catalyzed reactions. Also known as PMP. Now, notice PMP has an amine group here, which replaces the aldehyde group, which is the business end of the molecule. Now, in all reactions with PLP, this cofactor is actually covalently bound to the enzyme that uses it. Typically, there's a lysine in the active site of the enzyme. As you remember, lysine has an amine group, and this can form a shift base with the aldehyde. The reaction proceeds in 2 steps. First, we form a tetrahedral intermediate. As such, and then we form the shift base. So this will be the enzyme-bound PLP. And this is where all the PLP catalyzed reactions start. Let's take a closer look at the transamination reaction. Transamination reaction occurs between an amino acid and an alpha-keto acid. We have here amino acid 1, where we highlighted the amine group, and alpha-keto acid 2, as you can see, there's a keto group next to the carboxyl. Now, in a transamination reaction catalyzed by PLP, the amine group moves from the amino acid to the keto carbon of the alpha keto acid. And we obtain a new alpha keto acid and a new amino acid. So, in effect, the PLP catalyzed transamination reaction facilitates the transfer of the amine group from an amino acid to an alpha keto acid. Now, this reaction actually occurs in two steps. In the first step, the amino acid transfers the group to the cofactor itself. So, if you remember from the previous slide, the PMP contains an amino group, and that will actually contain this amino group that was taken from the amino acid 1. Now, in the second step, PMP will transfer its amino group to a different alpha keto acid to generate a new amino acid. Now, there are enzymes for virtually every single amino acid that can accomplish this first transformation, where by using PLP, they transfer the amine group and form an alpha keto acid and the PMP. Now, in the second part of the reaction, however, the alpha keto acid 2 is typically alpha keto glutarate or oxaloacetate. So in this case, not any alpha keto acid can function. Alpha keto glutarate or oxaloacetate. Now, let's take a look at an example. For example, glutamate is going to be our amino acid, and oxaloacetate is going to be our alpha keto acid. And in the PLP catalyzed transformation, we will obtain the alpha keto acid corresponding to glutamate, which is alpha keto glutarate, and the amino acid corresponding to oxaloacetate, which is aspartate. This enzyme that catalyzes this transformation is in fact ubiquitous, and it's found both in the liver, in the muscles, and it is in fact we can call it, depending on the products, we can call it aspartate transaminase or glutamate oxaloacetate transaminase. In fact, if we find this in the bloodstream, this enzyme acts as a biomarker, and it tells us about some damage that might have occurred in muscle or liver, which forced the cells to spill out their contents. This biomarker you'll often see as SGOT, serum glutamate oxaloacetate transaminase. And this is one of the biomarkers that are measured in blood tests that tells us about heart disease or liver disease. Let's take a look at the mechanism of the transamination reaction. And in particular, we're going to take a look at part 1, which, as we discussed before, the amino acid reacts with PLP to form an alpha keto acid and PMP. Here is our cofactor PLP, covalently bound to the lysine in the active site of the enzyme via a Schiff base. And here is our amino acid, the starting material. So in the first step, the lysine that forms the Schiff base with a cofactor is going to be replaced by the amine functionality of the amino acid, and it will form a new Schiff base with the cofactor. This starts with the amine group attack on the pyridoxal carbon to form a tetrahedral intermediate. And following an additional proton transfer, the lysine can be kicked off to form the new Schiff base. So far, we have started with the shift space corresponding to the PLP bound to the enzyme, and we now obtained a cofactor forming a shift base with the incoming amino acid 1. So this portion of the mechanism is called transamination, because we're starting with one imine and we're forming a different imine. Now, let's take a look at the alpha proton attached to the alpha carbon, which we're going to highlight here. This proton is now in between two carbonyl-like groups. Here is the carboxyl group, and here is the shift base group. So it becomes acidic enough that it can be removed by an active site base, for example, the lysine in the active site. This will generate a carbanion alpha carbon. This carbanion can only form because it is resonance stabilized. And indeed, the PLP ring system, it's highly conjugated and it's a good electron sink. For this carbanion, we can write, in fact, many different resonance structures. Let's take a look at one of them. This symbol denotes resonance structures. Notice in this structure, the positive charge on the pyridine nitrogen is now gone and highlights the fact that this is a good electron sink. And the ring now looks more like a quinone. That's why we call this a quinoid structure or intermediate. This quinoid structure shows us a glimpse into how the reaction will proceed, because the alpha carbon now it's doubly bonded to a nitrogen, which anticipates how this alpha carbon will become a keto group, and a product of the reaction will be an alpha keto acid. What happens? The quinoid structure can be reprotonated, but at a different place. For example, on the aldehydic carbon of pyridoxal. To highlight that these structures are in fact resonance structures, I'm going to put them in brackets. So let's take a look at what happened in this past couple of steps. So we had an alpha proton that was fairly acidic. It was able to be removed by the active site lysine. And then this proton came back to a different position. So all just happened in this couple of steps was a proton transfer. Now, looking at this intermediate, we can see that, in fact, the shift space or the imine of the PMP form of the cofactor and the alpha keto acid corresponding to amino acid 1. So via a hydrolysis reaction, these two can come apart. So in the first step, an activated water molecule attacks the alpha carbon, forming a tetrahedral intermediate. And one more proton transfer, and we can kick off the pyridoxamine form of the cofactor. And notice we obtain PMP and the alpha keto acid corresponding to the amino acid 1. This last step is in fact just a hydrolysis reaction of a shift base. Now let's take a look at the second part of the PLP transamination reaction. The part 1 left us off with formation of PMP. So in this second part, PMP will react with a new alpha keto acid to regenerate PLP and a new amino acid. In fact, this part of the mechanism, it's the exact reverse of part 1. Here is PMP and our alpha keto acid. In the first step, we're going to form, as we've gotten used so far, to a new imine between the keto group of the alpha-keto acid and the amine group of PLP. As usual, first we're going to get a tetrahedral intermediate. And one more proton transfer, and we can kick off the OH group to form the imine. Now, this portion of the reaction is, in fact, imine formation, which is the reverse of the hydrolysis step that we saw in part 1. Now, as you remember, there's an active site, lysine, which can act as a general base. And it's going to deprotonate one of these two protons on the pyridoxal ring. The reason we can form this carbanion here is because this negative charge is delocalized throughout the entire ring system. And let's show one important resonance structure. Which is none other than the quinoate structure we saw before. Just as before, the lysine, the protonated lysine, can now donate the proton on a different position, for example, the alpha carbon of the alpha keto acid. As you can see here, now the proton is on the alpha position, and now this starts to look more like the shift space formed by an amino acid with the PLP version of the cofactor. So from here onwards, we're just going to substitute the PLP, the amino acid bound to the PLP with the active site lysine in the transamination reaction that we saw before. So first, the lysine can attack this carbon. Forming a tetrahedral intermediate. And then, following some proton transfer, we can kick off the amino acid and generate the shift space corresponding to the PLP cofactor bound to the enzyme. So here we have PLP enzyme bound and the new amino acid 2. We mentioned PLP is a very versatile cofactor. So let's take a look what other reactions besides transamination can PLP catalyze. One interesting reaction used especially by bacteria is racemization. This involves taking an L amino acid, for example, L-alanine, and converting it via a PLP catalyzed reaction to D-alanine. This is an important reaction for bacteria as they incorporate D-alanine into their cell walls, which makes it very hard to recognize by the immune system and makes it very hard to digest by the host proteases. Let's take a look at a key intermediate in the PLP catalyzed reaction. As before, L-alanine is going to react with the PLP bound to the enzyme, and it's going to form an imine. Here we're highlighting the stereochemistry of the alpha hydrogen. And here is our active site lysine. As we've seen before, this alpha hydrogen is acidic enough that it can be removed by the lysine. And it's going to form a carbanion at this position. Now, this carbanion, as we've seen before, is able to form because the charge is in fact delocalized through the entire system of the pyridoxal ring. So for this structure, we can write a number of resonance structures, which we're not going to mention here. Now, this carbanion could be reprotonated, and here we had the hydrogen was pointing up on top of the plane of the page, but we can reprotonate it from the bottom, and that will change the stereochemistry at this carbon. So if that reprotonation happens from the bottom, we will obtain the shift base corresponding to the D-alanine. So by being able to generate this carbanion at the alpha position, the Plp reaction and cofactor allows the inversion of the configuration at the alpha carbon, converting the L-alanine to D-alanine. Now, another interesting reaction that requires Plp is decarboxylation. Here, we're looking at an amino acid, for example, glutamate. In a Plp catalyzed reaction, it can lose this CO2 group and form this molecule, which is called gamma amino butyric acid, or GABA. This is in fact a very important neurotransmitter, an inhibitory neurotransmitter that is required in the brain. So let's take a look how this decarboxylation is catalyzed by Plp. As always, as we've seen so far, the glutamate will react with Plp bound in the active site of the enzyme to form a shift base. Here is the shift base. Like that. Now, instead of deprotonating at the alpha position, the CO2 is activated to leave because it will leave behind a carbanion, such as that. And as we've seen before, a carbanion formed at this position can delocalize throughout the entire pyridoxal ring, and therefore it's stabilized and it can exist long enough. And we're not going to draw, but there, you can imagine there are a number of different resonance structures. Now, this carbanion, it gets protonated quickly by a general acid and will generate this structure, which is just the shift base corresponding to the gamma amino butyric acid with Plp. And now, from here, a transamination where the active site lysine will remove the Plp and free up the GABA products of the reaction. In this video, we talked about Plp catalyzed reactions. Plp is the cofactor that comes from vitamin B6. Here's vitamin B6, what we call pyridoxine, which is the molecule that we find in our vitamin pills. Now, in the body, pyridoxine forms pyridoxal, which is activated to form pyridoxal 5-phosphate, or PLP. And typically when it reacts, PLP is found as a shift base bound in the active site via a lysine. PLP is very important for transamination reactions, which are essential for the metabolism of all amino acids. We have seen the transamination reaction where an amino acid 1 reacts with alpha keto acid 2 and via PLP catalyzed reaction forms an alpha keto acid 1 and amino acid 2, essentially transferring the amino group from the amino acid to the alpha keto acid. The reaction occurs in 2 steps, where first the amino acid is transferred to PLP to form PMP, then PMP transfers this group, the amino group, back to alpha keto acid to generate a new amino acid. As we saw, the mechanism of transamination involves multiple steps. The first step is a transamination reaction, where the shift space that's formed between the active site lysine and the PLP becomes a shift space between the incoming amino acid and PLP. Next, we have a proton transfer, which is allowed by the ability of the PLP ring to stabilize a negative charge and moves the proton from the alpha position to somewhere on the PLP ring via a quinoid structure. And finally, the resulting shift base is hydrolyzed to generate PMP and an alpha keto acid. In the second part of the reaction, PMP now reacts with alpha keto acid to form a new shift base, and then the proton transfer happens in reverse via, again, a quinoid structure to generate the shift base corresponding to the PLP and the new amino acid 2, which via a transamination reaction would generate amino acid 2 and the PLP enzyme bound. Finally, we mentioned that PLP is very versatile and it can catalyze other reactions such as racemization, for example, switching the configuration of the alpha carbon from L-alanine to D-alanine, or decarboxylation, generating alpha-aminobutyric acid or GABA, an important neurotransmitter, from glutamate.